and the other one is now, and we are going to be talking about North American lakes. Next slide. As a reminder, we are a team of Harvard data science graduate students, and we wrote our thesis in collaboration with NASA for this part of the project with Dr. Bridget Vivien, as well as with our Harvard professors, Dr. Bruce Huang and Dr. Stephen Alston. Next slide. And in case you are interested in such a collaboration, this usually lasts a couple of months. The next cohort of Harvard data science students is starting about 2 months from now. So in case you are interested in collaborating with the next cohort, scan the QR code, follow the link, or email me directly, and I am happy to put you in contact with the right people. And with that, I am handing it over to Bridget, who is going to be talking about data collection.
Hey, everyone. So I was lucky enough to work with this amazing group of data scientists. And if you're thinking about doing the collaboration like Vivien was just talking about, I definitely suggest it. And if you have any questions, please reach out to me. It's been a really wonderful experience. So they're going to be talking about some data that I collected on some lakes in Wisconsin and Minnesota back in the summer of 2019. So there's 10 different lakes. I used the satellite data to kind of guide which lakes I was going to. They're going to get into details about how the lakes were similar or different, but that's how the lakes were selected. You can go to the next slide. I just have a few pictures to show what the data collection looked like. So Ryan Vandermeulen came along with me to get it started. You can look at these different pictures of the lakes and just see, looking at the color, that the different lake collection conditions were very different. So some are very, very green, some are more turbid. We had some young scientists getting involved, just taking pictures of a jar of water and with the station information, so we could just kind of compare at that very eye level. But then we were also taking a variety of more scientific samples as well. So you can show the next slide. This is some of the data that they were looking at. So we took discrete samples and filtered for chlorophyll. We had Secchi depth, and then I also had an ASD, so that's hyperspectral data that they looked at, and then we combined that with the Sentinel-3 data, the ULTI data that we use for cyan, and that algorithm is our CI-cyano, so it gives us an estimate of cyanobacteria in the water, and then also we use that to estimate chlorophyll. So that's just a little bit background on the data set. And then one more slide for me before we get into the exciting stuff. So when I got the email for Vivien, this is the state of the analysis. So this is where I was at last summer. And she said, hey, we were wondering if you might have a data set we can explore. And I said, oh yeah, I sure do. So I was really excited to collaborate with them. And so again, the team is Doris and Ronan, Vivien, and Walter. You're going to hear from all of them today. It's going to be great. And one exciting thing that's come about of this effort is that they're actually going to have this presented at the AI for Good, which is a UN summit that's happening at the end of May, so this month. So it's been like an amazing experience. They've done really great work. And I'll stop so they can take over.
Thank you very much. And with that said, we are going to be talking about 4 topics. I am going to be talking about the satellite to chlorophyll analysis. Ronan is going to be talking about the ASD hyperspectral data to chlorophyll, Walter about Lake Mendota deep dive, and Doris about time series with satellite data. So with that said, I am jumping right into satellite to chlorophyll. So as Bridget mentioned, we used the cyan data and matched it to in situ chlorophyll observations. What you can see in front of you on the left-hand side, it's a satellite image of Lake Koshkonong in Wisconsin. On the middle image, you can see the data that is coming out from the satellite from the cyan project. The yellow and green colors are basically representing the predicted chlorophyll coming out from this data set. And on the right-hand side, you can see a zoom in on Lake Koshkonong. In the blue circles, you can see where the in situ water samples were taken from. So in this specific project, we measured the cyan chlorophyll predicted data to the in situ chlorophyll data and did an algorithm of what is the best prediction between the two. Next slide. So as Bridget mentioned, there are 10 lakes in this data set, out of which we analyzed 83 samples. And one thing that stood out immediately is that these lakes are very different from each other. On the left-hand side, you can see a box plot. On the horizontal axis, there are the different lake names. And on the vertical axis, there is the chlorophyll as it comes out from the in situ analysis. And what stands out immediately is that there are high chlorophyll lakes and low chlorophyll lakes. The high chlorophyll lakes are highlighted in red, and then the low chlorophyll lakes highlighted in green. And what's interesting is that these lakes produce only exclusively low chlorophyll levels or high chlorophyll levels. We plotted this data on the map on the right-hand side. Obviously, the ones marked with red are the high chlorophyll lakes, and the ones marked in green are the low chlorophyll lakes. Next slide. After this, we took an even closer look of these different lake profiles, and we realized that these lakes, even though they are only within a 1-hour drive from each other, they can have very, very, very different lake profiles. Again, you can see high chlorophyll lakes and low chlorophyll lakes right next to each other. And taking a closer look of the profiles of these lakes, we could see that some of them are more like for fishing, for boating, for going to the beach. One lake is well hidden in the middle of the forest. So what it made us understood is that despite these lakes are so close to each other, every single lake is their own little ecosystem, and thus it is not surprising that they have very, very different chlorophyll levels. Next slide. So with this in mind, we built 30 different models to try to understand what is the best model that would translate the science satellite data into the in situ chlorophyll observations. We categorize these models into two categories. On the left-hand side, you can see these two categories. One is model complexity. We built both simpler models and black box models. And the other category is the location specificity, whether we included location data into the model, such as the lake name or this chlorophyll profile, or we did not include this data into the models. Then on the right-hand side, we plotted these 30 models. On the horizontal axis, there is the R-squared performance on the trained dataset, and on the vertical axis, there is the R-squared performance on the test dataset. So naturally, the best performing models are in the upper right-hand corner because those perform good on the train and on the test data. And a couple of things stand out immediately. Number one is that there are more red bubbles. Each bubble represents one model. That there are more red bubbles in the upper right-hand corner. This means that models that include this lake-specific data tend to perform better, which makes sense given what we saw earlier. The other thing that stands out is that simpler models work better. So you can see the empty bubbles are dominating the upper right-hand side corner. Furthermore, we took a closer look on the top two best performing models, which we highlighted in this green square. And the upper bubble basically includes the lake names, and the bubble right below it includes this chlorophyll lake profile, but not the lake name. And what it means that just by including this chlorophyll lake profile, the model has a very robust prediction. There is about an 87% R-squared performance on the trained dataset and about a 70% R-squared performance on the test dataset. So next slide. And here we plotted the predictive performance of this best model. On the horizontal axis, there is the actual chlorophyll as it comes out from the in situ dataset. And on the vertical axis, there is the predicted chlorophyll based on our model, based on the cyan data. You can see that the dots are very close to the 45-degree line. So there is a high correlation. Actually, the correlation is 83%. So this is about the chlorophyll 2 the satellite data to chlorophyll algorithm. And Ronan worked on a similar data set, but he used ASD hyperspectral data.
Thank you, Vivien. So can you go to the next slide, please? So like Vivien said, this part of the work uses a different subset of the same data set that Vivien worked on. So instead of using satellite data, I worked on hyperspectral data using a handheld analytical spectral device, or ASD for short, which is what you see in the middle. The advantage of hyperspectral data is that it captures more detailed information across wavelengths than satellite data, which expands the research that can be done with it. This data is also closer to what is available from the PACE satellite than to the data of previous satellites. On the bottom left corner, you can see the measurements for Lake Tashkent on July 9th. And it is noticeable that there are differences in the measurements, particularly for the one with the highest level of chlorophyll. It is also interesting to see that the measurements can vary quite a lot over the same day and in different parts of the lake, which is something that Doris will cover in more detail later. Next slide, please. So for this particular wavelength pattern, which I showed, which peaks around the 700 nanometer range, it is consistent across the observations with higher chlorophyll values, which is something that caught our attention. At first, we wanted to identify exactly which wavelengths were the most impactful for predicting chlorophyll, but the low observation count, the high number of wavelength measurements, and the wide range of chlorophyll, which Vivien explained in the different lake profiles, we had to change our approach. Instead, we focused on predicting chlorophyll as best as possible. Next slide, please. So we took several steps to get the best possible results, the first of which was to test both regression models and classification models. In the regression case, the goal was to predict the numeric level of chlorophyll. And in classification models, we tried to evaluate whether or not the level of chlorophyll was above or below 12 micrograms per liter, which is a WHO threshold separating lower and higher alertness levels for monitoring recreational waters, with different plans of action for dealing with the matter depending on the classification. To check if the performance results were consistent on unseen data, we used cross-validation. Lastly, for the regression case, we plotted the prediction intervals for the models to visualize how widely they vary. Next slide, please. These are the results for the regression models. On the left-hand graph, we have the mean R-squared for cross-validation on the y-axis and the R-squared for the test sample on the x-axis. The right-hand graph shows the prediction intervals for the best performing model, which was the one with the best cross-validated with mean R-squared, the one with the mean square around it. This model, support vector regression, had an average of around 75% in cross-validation and around 86% on test data. The lower cross-validation metrics compared to the test data in the graphs was the result of the presence of the very high chlorophyll observations, which you can see on the right-hand graph. However, even for these observations, the prediction bands all fell within the levels of what the WHO would consider of higher alertness, suggesting further investigation of the water for cyanobacteria and toxins. Next slide, please. Now, these are the results for the classification models. On the left-hand side, we have the same type of graph as before, except this time we are using accuracy as the metric. We can see on the right-hand graph the predictions for the best performing model, which was K nearest neighbors, as well as the prediction of the regression model, which is the orange dots. The green dots on this graph are the predictions that the algorithm for classification got correctly, and the single red X is the one that it got wrong for this test sample. The specific level of chlorophyll for their observation is 10, so right on the threshold, which may have explained the error in prediction. You can also see that the regression model was very close in predicting the actual values for this test sample, and these results are very likely to improve if these techniques are applied with more data. Next slide, please. Now, there are important risk factors and ethical considerations to discuss associated with these classification models. Take a look at Lake Tashkent on the left. You can see that there are hotels, beaches, wildlife area all around the lake, and these areas are all directly affected by the quality of the water in the lake. Ideally, our models would be able to correctly predict where phytoplankton levels are too high or too low 100% of the time. These are the true positive and the true negative rates. However, no model is perfect, so we need to be especially mindful of the false negative and the false positive errors. This is important because of the serious effects of high phytoplankton or high cyanobacterial levels on human and animal health, as well as the economy. When we have a false negative, our model predicts a low level of chlorophyll when it is actually high. So this can result in serious illness or death to humans and animals that interact with this water. On the other hand, false positives occur when we predict that the water has high chlorophyll values when they are actually low. This can result in needless economic harm if bodies of water, beaches, and resorts are closed unnecessarily. There are some caveats to this modeling approach. Since the predicted variable is chlorophyll and not cyanobacteria, the actual impact of high phytoplankton levels may change depending on the type of phytoplankton that is predominant in the water. Beyond cyanotoxins, algal blooms can block sunlight and cause oxygen depletion, which is something that other data sources cover and Walter will soon talk about. Next slide, please. Now, just 1 second. It is for those concerns that it is important to have a clear understanding of the capabilities and limitations of our models. And for that, we need to look beyond mere accuracy. The charts you see show different metrics for the models, but I would like to bring your attention to the second one from left to right, which is recall. Recall is the true positive rate, which is a measurement of how well the models can accurately predict the high chlorophyll cases. While you could just create a model that predicts high level of chlorophyll 100% of the time, which is what the majority predictor does, the blue bar, it would get all of the low chlorophyll cases wrong, which is what we see on the graph for specificity just on the right, or it is the graph for true negative rate in the way. In this case, we can see that the models perform better in general when predicting higher levels of chlorophyll than lower ones, which is acceptable considering the risks. And now we're going to go to the next slide with a small change of gears. We are switching gears and we're looking at a different problem, using apparent visible wavelength, or AVW, to predict chlorophyll. AVW is an algorithm that uses the weighted harmonic mean of the wavelength measurements and outputs a single number, which is the main wavelength value that represents the dominant color in the water. Using this technique, we can get this single number, and we can model it to predict the levels of chlorophyll in the water. We had two data sets, two numbers that were calculated using this AVW algorithm. The visible AVW that uses the weighted the band of light that we can see around the 700 to 400 nanometer range, and the CI cyano AVW area, which is that red one between 620 to 710. We can see that the visible AVW just narrowly misses the peak at the 710 nanometer range, and we believe that affected the performance of the models that we compared. So next slide, please. So these are the results for the linear regression model we had. On the left-hand side, we have the cross-validation metric box plots with the mean absolute errors at the bottom. The smaller the errors and the tighter they are, the better the model. And we can see the CI cyano AVW calculation outperforms the one with visible AVW, and likely because it didn't take into account the information in the 710 nanometer range. And as you can see in the predictions, with a 64 train test SR squared, which is a good performance, the same issue with the regression model from before happens, in which the errors in prediction happen for the higher levels of chlorophyll. And with that, I finish my part of the presentation, and I take it over to Walter.
Thank you, Ronan. The goal of my part of the project was to deepen our understanding of freshwater phytoplankton using in situ data from Lake Mendota, located right next to Madison, Wisconsin, so in a continental climate with cold, snowy winters and warm summers. This climate means both humans and phytoplankton use the lake differently, depending on the season. I'd like to show some examples of what we found interesting about it. Next, please. The analysis is based on data gathered near the deepest point of Lake Mendota using different devices. For example, variables like chlorophyll and phycocyanin were recorded by submersible fluorescence sensors attached to a buoy at the depth of 1 meter. Water temperature was recorded at different depths using a thermistor chain that remained in the water for most of the year during 17 consecutive years. Next, please. We were interested in water temperature of the mixed layer in particular, so to identify the thermocline depth, we analyzed the lake's thermal profile. Each line on this plot represents water temperature at the same time at different depths. Lake Mendota has a typical four-season lake profile, so as we go deeper and deeper, all the lines converge to 4 degrees Celsius as the temperature of maximum density water that always sinks to the bottom. What's going on closer to the surface depends on the season. During the summer, shown here as orange color, our data show strong thermal stratification. For the winter, represented by blue lines here, we didn't have a lot of data, but we can still see some blue lines leaning to the left as an indication of inverse thermal stratification. Some green lines, being vertical, show that Lake Mendota becomes isothermal during the spring, with the same temperature and density throughout the entire water column, which makes it relatively easy for the wind energy to mix water deep in the lake, and so lake turnover takes place. Next, please. To identify different types of algae, we analyzed chlorophyll and phycocyanin levels expressed in relative fluorescence units. Exploring time series plots like top left here, we noticed indication of algal blooms that don't look very regular. Both chlorophyll and phycocyanin have shown very different magnitudes for some of the years compared. Some pattern of bloom is associated with seasonality. The plot on the right shows bivariate analysis of two variables, chlorophyll and phycocyanin, as representing different types of phytoplankton. Blooms indicated by high chlorophyll level happen usually in the spring, so right after the lake turnover, while the highest level of phycocyanin can be observed especially during the summer when water temperature is the highest. Next, please. First, first results of exploratory data analysis of dissolved oxygen raised some data quality concerns that eventually turned out to be interesting discoveries about different types of diversion from ecosystem equilibrium associated with algal blooms. First, when algae bloom, the high photosynthetic activity leads to oxygen supersaturation close to the surface. And this is why the histogram on the left shows large part of the distribution of dissolved oxygen saturation being above 100%. Looking at this histogram, we were concerned about the huge peak at zero, but further investigation revealed that this is not a data quality issue, but an indication of a natural phenomenon in Lake Mendota, the formation of a dead zone pretty much every summer the data cover. For example, year 2020, shown on the right plot, with time during the year on the x-axis, depth on the y-axis, and dissolved oxygen level color-coded, the dark color showing when and how deep oxygen goes to zero. Apart from algal blooms blocking the sun rays from reaching deeper levels of the lake, high temperature-induced stratification stops mixing different layers, and in the dark, isolated from air hypolimnion, oxygen is consumed by microbes and goes to zero. Another thing we noticed on that right plot is the spring and fall turnover, replenishing dissolved oxygen in the deepest levels of the lake. Next, please. Because the harmful effects of dead zone depend on water temperature, we wanted to estimate the long-term trend of this variable. We found it interesting from data science methodology perspective. When we look at the time series plot in the left upper corner, we can notice some missing data, irregularities. Generally, at the beginning, temperature was recorded mostly for the summer. Then, after 2016, more colder months measurements were included. For this reason, simple OLS trend estimation result suggests that Lake Mendota is getting cooler just because colder months are underrepresented more at the beginning of the time series. But we get completely different result when we do it the way explained graphically in the upper right corner. where estimation is not impacted by seasonality. First, we group all the data by week number and then estimate separate trend line for each week during the year, taking data from all the 17 years available. This way, we would theoretically get 52 trend lines because of 52 weeks during the year. In reality, we get fewer just because we don't have enough data for each week, especially during the winter. The bottom left plot shows results of trend estimation for the weeks between March and November, mostly positive, especially during the summer weeks. Bottom right bar plot shows the final results after aggregating all the group results. It also compares what we obtained to a published paper result estimated based on over 100 lakes from different continents. So Lake Mendota gets warmer by 0.28 degrees Celsius per decade based on the average for 17 years analyzed here. Next slide, please. So Lake Mendota Institute data analysis shows how different types of phytoplankton and effects like dead zone, which is very harmful to aquatic life, are influenced by seasonality, but also long-term changes of temperature. So monitoring and predicting algal blooms are becoming more and more important, and Doris has done a great job building tools to do this. Thank you. Doris, you are muted.
Can you hear me now? Yes?
Yes.
Sorry about that. Okay, let's start again. So the main objective of this scope is to see how satellite data from the SCIEN project can be leveraged for cyanobacteria analysis and forecasting. First, let's take a look at data processing. The data we use is the level 3B daily data from the NASA Ocean Color website. We convert the data into tabular format, add geolocation information, and stack them together to form a time series dataset. The pre-processing steps are simple, but we did face two major challenges. First, the data files are big, and it also takes a lot of time to download and process them. For example, to process the data for the entire North America for 1 year, it will take about 4 hours, and it will take also over 80 gigabyte of storage. To address this issue, we highly recommend that to store the SCIEN data in a SQL database server so that users can extract the data for the exact time and location that they need without having to process the entire data files. Now that we have the pre-processed data, what can we generate we can generate some insightful plots. First, let's take a look at the satellite data for Lake Mendota, the same lake that Walter just presented in the last scope. What we have here on the top left, we plotted the number of observations available from 2016 to 2024 by geolocation, so longitude and latitude. The blue dots on the edges of the lake indicate low data volume, and the red dots in the center indicates high data volume. On the right here, we also plotted the average cyanobacteria level of the lake for the same time frame. So the red dots here represent high concentration of cyanobacteria, and the blue dots indicates low concentration. It is interesting to see that the high concentration, high cyano level tends to happen on the edges of the lake, but also given that we have low data volume on the edges, these readings might be a little bit biased. Another observation that we have is that the northern tip of the lake tends to have lower cyanobacterial levels, as well as the southeastern side of the lake here, indicated by the bluish color. Comparatively, the west side of the lake generally have higher cyanobacterial level, indicated by the yellowish color. Down here, we can actually visualize how the cyanobacterial level changed month over month, as well as the trend over the past 8 years. Next, we look at seasonality. The top plot here shows the average cyanobacteria levels for each month. The x-axis are the months from January to December, and y-axis are the years. The color indicates how high the cyanobacteria level was for that particular month. Red is high and blue is low. The bottom row of this plot shows the average values of each month. So we can see that cyanobacteria levels tend to peak in the summer from May to October. You will probably already noticed this red box here that shows that the cyanobacteria level was extremely high in June of 2018. When we look at this spatial analysis of that month, you can see that the west and south side of the lake was impacted the most. On the right here is a satellite data image of the lake around that time, showing a bloom on the west side of the lake, which matches the spatial analysis we have here. And the bottom here is just a news article that talks about the algal bloom that happened during that time. It mentioned that the bloom was caused by heavy rain washing manure into the lake, causing high level of phosphorus, which helped with the algae growth. The second plot down here in the bottom shows the number of data points available each month. We can see that there were significantly less data available in the winter, which matches with what Walter just mentioned in the previous scope. Next, we move on to cyanobacteria forecasting. So here again, we use only cyan data, satellite data here. No external factors such as temperature or mineral levels are included in the model. We actually tested a couple of time series models and finally decided to use the SARIMA model. SARIMA stands for Seasonal Autoregressive Integrated Moving Average. It is a parametric statistical approach that works by predicting future values of a time series based on the parameterized values of the past time steps. In the interest of time, I won't go into too much detail on the methodology. But I would mention that another great feature of this approach is that we are able to separate seasonality from the real trend. Here is an example for Lake Mendota again. You can see that the actual trend when the seasonality is separated, plot separated. Okay, now how does the model perform on real data? On the top, this is a plot of the average weekly cyanobacteria level of Lake Mendota from 2016 to 2024. The blue line is the actual data, and the orange line are the model predictions. You can see that for the most part, the orange line is very close to the blue lines, but sometimes it kind of misses the mark, like this one here in mid-2019. We also assess the model performance by its ability to classify low, medium, or high cyanobacteria levels. We can see that the model performs reasonably well with over 85% accuracy for both training and test data. But sometimes there is a bit of a delay, just like this one in around 2023 of May of 2023 to June. The reason could be due to irregular activities that not being captured by the model. As you can see that this spike in May 2023 actually didn't happen at the same time in 2022. And this is one of the main drawbacks of not considering external factors in these forecasting models. Another thing to note about this type of time series models is that they tend to be more accurate when predicting values for near future and less accurate when we try to predict further ahead in the future. So finally, I'll showcase an interactive dashboard that combines our findings. Here on my web browser, you can see basically similar plots we showed in a couple previous slides. On the left is the number of observations for Lake Mendota. On the right side is the average cyanobacteria level of the lake. Here, it's a little bit more interactive because you can also choose the date range you want to analyze the data for, such as if I want to just look at past 12 months, I can change that. And now this is just the data based on the data for the past 12 months. And when you hover your mouse onto the plot, like there is a window that will pop up to show you exactly the CI signal values for that particular location. And also here is the number of observation for that location. There is also the trend plots like here, and also the seasonality plots down here. We also have a separate visual for the prediction. So similar to what we showed in the slide, the blue lines are the actual data here, and the green lines are the predictions, basically. And then the last view is the seasonality and trend decomposition. So all of these are written in Python. And the code and the methodology are all shared. I put them all on GitHub. So feel free to take a look, and if you're interested, and I welcome any feedback or comments. So with that, it concludes our presentation today. Thank you very much for your attention. Then we can go on to Q&A section.